Hello, everybody. Now that you know what transcription and translation are, I want to talk to you about the genetic code. The question we have at this point is, how are the instructions for assembling a chain of amino acids encoded in the DNA? We know that there are 20 different amino acids in proteins, but there are only 4 nucleotide bases. So how can these 4 bases be used to represent 20 different amino acids? That's our question. If the bases are read one at a time, A, C, G, or T, that would only provide for 4 codes, but we need 20 codes for each of the amino acids, so that doesn't work. If the bases were read 2 at a time, so for example they were read AA, AT, AG, AC, etc., there would only be 16 possible codes. Well, that's not enough. So it seems logical to suppose that the bases must be read 3 at a time, because that would give us codes of AAA, AAT, AAG, AAC, etc. And it would give us 64 total codes, which is more than enough to account for the 20 amino acids. It turns out that indeed the genetic code is based on a triplet code. We read the bases in the nucleic acids as triplets. Each set of 3 bases is called a codon. That's a vocabulary word you need to know. The code is non-overlapping. In other words, the sequence in DNA of AGT TCA would yield 2 codons. The first one is AGT and the next is TCA. For example, AGT at a particular point in a DNA strand results in the placement of the amino acid serine at the corresponding position of the polypeptide to be produced. So that codon AGT in DNA will ultimately lead to the amino acid serine being put into a polypeptide. Let's take a look at this in a picture now. And you have this picture. It's one of the diagrams I've given to you, so you can follow along on that. Here's the idea. Here is a chromosome, and you can see in this section of this chromosome there are 3 genes, labeled gene 1, 2, and 3. And here in the middle of gene 1 is a sequence of nucleotides, which are ACC, AAA, CCG, etc. And during transcription, they're going to be turned into their complementary nucleotides, but they're going to be ribonucleotides, not deoxyribonucleotides. So they're going to be RNA. U, G, G, because U is complementary to A, G is complementary to C, G is complementary to C, U, 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 G, G, C, U, C, A. And then that mRNA will travel to a ribosome in the cytoplasm and translation will occur. And the UGG codon will tell the ribosome to put a tryptophan there. The UUU codon will say put a phenylalanine next in the polypeptide chain. The GGC codon will say put a glycine next, and the UCA will say put a serine next. That is how the genetic code works. Now you might be wondering, how did we figure this out? Well, it was a painstaking process that took several years. But what someone did was they made a piece of mRNA completely out of U's, and then they placed it in a test tube with ribosomes and a single amino acid. And they did that 20 times, 1 for each amino acid. And what they found was when the amino acid phenylalanine, PHE, was placed in the tube, a polypeptide was made. But in the other 19, no polypeptide was made. So they knew that the code UUU meant put a phenylalanine in the polypeptide. And so they used this kind of experimentation to figure out the codes for all the amino acids. And they slowly worked it out. Every possible base sequence has a meaning in the genetic code, and here now is a picture of the genetic code. And I want you to know how to read this. The left-hand side shows the first RNA base. The top shows the second or the middle RNA base. And the right-hand side shows the third RNA base. And you can see here it is UCAG. So let's look at the upper box. The code UUU codes for phenylalanine, U, U, U. The code U, U, A, as you can see there's the A, codes for leucine. Let's look at another one. A, U, right? So A, U codes for asparagine, I think that is. And the code G, C codes for glycine. So you should make sure that you understand how to read this code given a sequence of letters. You will also notice in here that there is a methionine or start codon here. And I'll explain that in a second what that means. And that there are 3 stop codons. So I also want to explain them. A couple of important things to know about this code. The stop codon causes the cessation of translation. So if the codon UAA comes along, the polypeptide will no longer grow, no matter what's on the other side of it. So it always causes the end of translation. The AUG codon is always at the beginning, but it doesn't just mean start, it also puts a methionine in. So it turns out that almost all proteins, their first amino acid is methionine. So that's a second important thing you need to know. Third thing is that the genetic code that you see here is a messenger RNA code. This is not what the DNA looks like. To get to the DNA, you have to take the complements of these codons to see what the original DNA looked like. And we'll see about that in a little bit. I'll give you some exercises where you will see how that works. Another thing you will notice here is that some amino acids have many codes. For example, serine has 4 right here and 2 right here, so it has 6 codes. Whereas others have very few codes. Methionine has a single codon, AUG. And finally, you will also notice that for many of the amino acids, the third codon does not make a difference. If the first 2 are CC, it doesn't matter what the third one is. A proline is going to be put there. If the first 2 are AC, it doesn't matter what the third one is. A threonine is going to be put there. If the first 2 are GC, an alanine is going to be put there. So those are the important things for you to know after looking at this table. And that information is all contained on this slide. So now that you understand the genetic code, I want to go on and look at translation. And we're also going to learn about the third RNA, the transfer RNAs. Amino acids that are floating around in the cytoplasm, in order to be translated, have to be attached to the third kind of RNA, the transfer RNA or the tRNA. For each amino acid, the tRNA is unique. So every amino acid has unique tRNA. And each tRNA has an anticodon that is the complement of the codon on the mRNA. For example, if the mRNA codon is AUG, then the tRNA anticodon will be UAC, because UAC is the complement of AUG. Why does the tRNA have to have an anticodon? Because that is how the proper tRNA will pair with the messenger RNA, as we're going to see in a second in pictures. So let's see how this all works. So first, this is what a tRNA looks like. It's got these 3 arms on it, these 3 loops. One of them is called the ribosomal site, and that's where it's going to attach to a ribosome. Another is called the anticodon, and you can see that's here. And so GGC is the anticodon because it's the complement of the codon CCG. And then the enzyme site we're not going to worry about much in this course. But at this end, the carrier end, the amino acid is attached to the transfer RNA. Now, let's think about the ribosome for a second. The ribosome has 3 important sites: a tRNA attachment site, a polypeptide site, and an ejection site. So let's take a look at those. Here's a ribosome. You can see the large subunit and the small subunit, and you can see the mRNA is between the 2. And now there are the 3 sites. Here is a tRNA with its amino acid about to attach to the codon. Its anticodon will pair with the codon. So it is bringing in the amino acid glycine. And then Here is the polypeptide site in the middle, and you can see here is the growing polypeptide. The last amino acid to be added was phenylalanine. And then here is the ejection site. Here is a spent transfer RNA that is going to be ejected back into the cytoplasm, where it will pick up an amino acid and be able to participate in the translation process again. Now, translation is a dynamic process, so here are the steps. First, a tRNA with its amino acid attached arrives and attaches to the ribosome at the attachment site. How does it do that? Via hydrogen bonding with the mRNA transcript. In other words, the complementary basis hydrogen bond. Second, the new amino acid is added to the existing polypeptide by the enzymatic action of the ribosome. So the ribosome acts like an enzyme and attaches the new amino acid to the existing chain. And then after that, the entire polypeptide is now held by the tRNA at the attachment site. So next, what's going to happen? The whole thing is going to shift over, and the polypeptide is now in the polypeptide site. And at the same time, the spent tRNA is going to be ejected. And then when a new tRNA arrives at the attachment site, the process starts over. Let's take a look at that in pictures. So here it is. So we'll start at the top here. Here is a tRNA at the polypeptide site with a growing polypeptide chain. The next codon hydrogen bonds with the anticodon of the tRNA that holds the next amino acid in the sequence. So that's going to come in here and bond. And then this whole thing is going to be shifted onto this tRNA, and that takes energy. GTP, like ATP, is a cellular form of energy. So you can see what's going to happen here. The whole thing gets transferred over, and then now you have the old spent tRNA at the polypeptide site and the newly lengthened chain at the attachment site. And then it takes energy and the whole thing shifts over, and in the process, the tRNA is ejected, and then the process starts over again. So that's it for today. I hope what you understand is that the genetic code is a triplet codon, which means every 3 bases in a messenger RNA code for a single amino acid. The code is non-overlapping. There are 64 total codons, of which 3 are stop codons, which means they will stop the creation of a new polypeptide, or the translation will be brought to an end. 1 is a start codon, but that always puts in the amino acid methionine. So methionine is the first amino acid in virtually every protein. Then after that, I hope you understand how translation actually occurs on the ribosome, that the ribosome has 3 sites: an attachment, a polypeptide, and an ejection site, that the ribosome forms around the messenger RNA, that the tRNAs and their anticodons hydrogen bond to the messenger RNA, and that slowly over time you build a chain of amino acids, the so-called polypeptide. Don't forget that polypeptide will be subsequently processed in the Golgi apparatus, folded into the right shape, perhaps folded around other polypeptides, perhaps other atoms will be introduced, and you will get a functioning protein. Until next time, you should go outside, take a look at nature, ask biological questions, bring them back to class, and I'll see you soon.